Okay. Hello everyone. I am the PMRFTA for this course, Introduction and Advances in Omics. My name is Shomo. I am a second year PhD student at ISC Bangalore. So I will be taking this this TA sessions every Tuesday, 6 to 8 p.m. So how I do it is basically for the first 1.5 hours, we'll discuss about the topics that are covered in that week, a little bit of maybe something more if I have anything else to discuss on it. And after that, we'll discuss like uh, assignment questions, old assignment questions. What's the like last half an hour, 45 minutes of the session, or last 1 hour of the session, depending on the content. So uh, yeah, um, please try to join these sessions. They'll help you solve the assignments. And I would personally would want to know like what kind of backgrounds all of you come from. So it would be good if you could like probably unmute yourself and tell, or maybe write in the chat box if you have if you can do that. So it'll give me a better idea about like whom I am teaching. So uh, if you can, please write in the chat box or just unmute yourself and let me know. Uh, so this is an 8 week course. So the today's uh, part will be basically mostly your uh, introduction to what omics is, different types of sequencing. I'll not go into the details of the history of omics. I'll probably more focus on the technologies and like the, the chemistry behind that. So yeah, been seeing a couple of people from BSc in microbiology, BSc in botany. Okay. So yeah, anyone from any mathematical background or engineering background? Anyone? Okay. Okay. Yeah, one more thing is these lectures will be recorded and I will be uploaded them on YouTube maybe a day later after that. And you can, I think you'll have access to the sheet where all the YouTube links will be provided. So you can, if you miss any of these classes, you can go back and check out the recorded lecture. It will be available on YouTube, and you can check it out if you have missed the lecture. Okay. So let's get started then. So I'm assuming most of you have biology backgrounds, so I'll go about like that only. Okay. I hope you can see my screen. Can you just reply once in the chat? Can you see my screen? Okay. Cool. Yeah. Okay. I'll be talking about the sequencing types, and like mostly these type of sequencing are not is not done today, because we have much better technologies. We have better right now we have like long read technologies as well, so those are much better. So we'll start from the initial type of sequencing. The the first type of it was Gilbert sequencing. It was also discussed in the week 1 lectures. So yeah, this was a sequencing method published in 1977. So around 50 years almost from today. And so this allowed purified samples of double-stranded DNA to be used without further cloning, and you could sequence uh, those DNA samples without much of an issue back then. So this was a very big thing that came out at that point of time. The basic idea behind this is basically where the cleavage of the bases occur in the whole DNA molecule. So you have the DNA molecule, which is a string of your ATGC base pairs, and out of that, various combinations will be present in the DNA, and what combination does this particular technology use of is basically these four type of combinations. So you have a G, you have a plus G break, you have a C break, and you have a C plus T. So this finds these type of combinations in the DNA and tries to cleave it. And then after that, after the cleavage, you run it through a gel, and you see bands of different of these different parts of the DNA inside the gel. You can visualize it. Yeah. So these fragments in the four reactions are electrophorezed side by side in these gels for size separation. And to visualize the fragments, the gel is exposed to X-ray film for autoradiography, and it yields a series of your dark bands, each band corresponding to a radio-labeled DNA fragment, and from which you can infer your sequence. So yeah, so this is the principle. First, the two DNA strands are separated, and then the labeled DNA is distributed into four samples in four separate tubes. And each sample is subjected to treatment with a chemical that specifically destroys one, either G or C, or two bases of like A plus G and T plus C in the DNA. So four combinations, four different types of enzymes that can digest the DNA in four different types of manner and bring these fragments. And then uh, it results in the formation of a series of labeled fragments of varying length. And the actual length of the fragment depends on the site at which the base is destroyed from the labeled end. Thus, for example, if there are C residues at position 4, 7, and 10 away from the labeled end, then the DNA that specifically destroys C will give a labeled species of 3, 6, and 9. It's basically just the so if you're cleaving the base at fourth position, you will get it till third position, the sequence length. Yeah. Yeah. Then the labeled DNA is obtained. DNA fragments are obtained in these four different tubes with four different enzymes, and then they are subjected to electrophoresis when side by side, where you can visualize these bands. So yeah, this is a visual representation of that. These are the four combinations you have. The gel. I hope you know how the gel electrophoresis works. The higher the mass of the fragment, the less traveling it will do. So uh, these are your on the top are your longer segments, and on the bottom will be your shorter segments, and there'll be a kind of a gradient of the length of the segments depending on the gel, basically how long your gel plate is. Depending on that, you'll have longer to shorter fragments distributed in the gel plate. So sorry. Okay. Yeah. So this is the principle behind Maxim Gilbert sequencing, but then we moved on to something called Sanger sequencing, which is also around that time, which is 1977, and this person Frederick Sanger, he won his second Nobel Prize for the invention of this technology, and this is a technology that is still used till date in some labs where you have to do low cost sequencing. You have to find sequence of a small DNA fragment. People go for Sanger sequencing till date, but it was very widely used till mid 2000s. After that, the next generation sequencing technologies came. But before that, this was a very popular and maybe the best way to sequence DNA fragments. Uh, so the principle behind Sanger sequencing is something called sequencing by synthesis. So basically, you have a strand of DNA and you want to know what is the sequence of the DNA. It's basically you sequence uh, its complementary strand to find what the other strand is basically. So yeah, this is the sequencing by synthesis technique, and basically in next generation sequencing, say for example in Illumina sequencing that we use today, it's basically a massively parallelized version of this Sanger sequencing technology actually. So in Sanger sequencing, so you have one well, inside that you have a DNA fragment, and then you synthesize the complementary DNA fragment, and then you try to make out what the sequence is. In Illumina sequencing, this technique is followed across multiple wells, hundreds of wells, where you can sequence multiple of these DNA fragments together. So it's kind of a more parallelized version of sandwich sequencing, but the core principle is similar, but we have a lot more like we have come a lot ahead in terms of technology and chemistry of these techniques. So uh, yeah. Yeah. If you want to sequence the DNA, you need DNA polymerase, and that is the enzyme that is used in this case in Sanger sequencing. And they use something called DNTPs to synthesize this new strand. And basically, these DNTPs are dideoxynucleotide triphosphatase, which is another modified version of DNTP. So DDNTPs are also used to find where the termination procedure has occurred in the sequencing, right? So the idea is as I was saying, is sequencing by synthesis. So it takes advantage of the fact that DNA strands, which are normally double-stranded helix form, split apart uh, during cell division or mitosis, and each strand is copied separately, replication of DNA. And this was the idea behind strand sequencing. Just a second. Yeah, yeah. So the idea is that DNA polymerase will naturally use these DNTPs to, if you have a single stranded DNA inside a test tube or a well, and if you put DNA polymerase enzyme inside that, and you have a lot of these DNTPs, so this DNA polymerase will take each of these DNTPs and start replicating the DNA, normally how it is done in a cell. Yeah. Yeah, so these DNTPs will be naturally used by this DNA polymerase to synthesize a new strand, and the synthesis process will occur very quickly, and it makes it hard for to make any sort of measurements of what base is getting added at at a base level. So there's this strand, and then one after another you're adding the base, so you do not know what base is getting added uh, if you just put the DNA polymerase and you put the DNTPs, it will just add it very quickly, and it is very hard to capture this. So one way to overcome this is basically trying to find out a way to terminate synthesis at your like whenever you want. So you want probably two bases to be synthesized, and then there is a termination, you want it to be terminated. So if it's like TTA, so the A complement will be T again. So the third T will have some sort of the base will be somehow modified so that after that no more bases can be added. So that is how science sequencing works as well. Yeah. This DNTPs can attach a DDNTP to the sequence just like a DNTP. So DNTP is your normal nucleotide triphosphate that is part of the DNA. The DDNTP is a modified version of that. So if a DDNTP is added, it will not sequence further, synthesize further. But if a DNTP is added, it will synthesize further. So basically, this DDNTP will halt the replication of the DNA molecule. So this is how it looks like. So you have a sequence, this is a single-stranded DNA. Then you have a lot of these DNTP molecules available uh, inside your culture or your well or your test tube, whatever. Uh, some of them will be DDNTP. So the star marked molecules are your modified DNTPs, which will act as terminating signals. So the proportion of these molecules are very low inside how it is designed is very low to be kept inside your experimental environment. So it is around like 1:99 ratio around. So if there is one DNTP present against that, there will be 99 almost DNTP present. So you add this single-stranded DNA, this set of your DDNTPs and DNTPs, and then you add the DNA polymerase enzyme. Again, if you remember DNA replication, it requires a primer to start the replication. So you also need to add a primer according to whatever sequence you're adding. So this primer will get added, and then the DNA polymerase will start to synthesize the rest of the strand. So in this case, it synthesized. So this was the primer TATG. till here. Then it added two more bases, and then it got terminated because the the next base that it added was a DDNTP. In another case, it got terminated at the second last position because there was a A there, and a T that was added was a DDNTP. But in most cases, it will complete the whole sequence because the proportion of bases that are DDNTPs are very low inside your experimental system. So in maximum cases, 98% cases, it will form the uh, whole molecule, the whole double-stranded DNA. But in some case, in very few of these cases, 1%, 2% cases, it will terminate before that because of the DDNTPs present. Now, how often do you want this to happen will depend on the concentration of DDNTPs inside your experimental procedure. Right now, once this has happened, this actually is a signal to tell you that if a T has been added here and no further sequence has been added to that, that means there has been a termination after the T, so that means there is a here. So now if you have the information, the data for at all the positions that there were probably DDNTPs added, and if you have all the combinations, so for example, you have a combination of so TATG is your primer. Let's consider that. After that, if you say, for example, you had a CDDNTP, you had added this here. Uh, it, After that, this part will not be sequenced. So you know there's a CDDNTP here. So the opposite base position will be G. Similarly, if you have a GDDNTP here, the opposite base position will be C, and so on. So if you have breaks at every base level, you can actually reduce the whole sequence out of it. Yeah. And so if you repeat this process in parallel using other DDNTPs, and you can get at every position of the DNA sequence, you will have a DDNTP added, and you'll know which DDNTP has been added, and uh, you can just infer the base that is complementary to that. So at every position, you have the termination sequence. So at every position, you know what the complementary strand is. So that is the general idea about how Sanger sequencing works. So this is basically a workflow of that. So you would want to first replicate the DNA in multiple copies. So you use polymerase chain reaction for that, and then you need to like separate the two strands of the DNA. The best way to do is using heat. So you heat the sample and the double-stranded bonds break, and it forms single-stranded DNA. And then you add this floating DNTPs with a few modified DNTP, and then you add a primer because you need the primer for replication initiation, and then you add in parallel you do it for other types of nucleotides as well, so that you can have a mass of these, a collection of these half synthesized DNA strands. From that, you can infer the whole sequence. Right now, this is actually a very intuitive way to solve this problem, but there are certain limitations to it. So, firstly, the sequence length. Sorry, I have to stop every time because people are coming in and I have to admit them. So sorry for that. Yeah. So yeah, it's a very intuitive way to do it, but still there are limitations to it. First is the sequence length. So Sanger sequencing mostly cannot be done above 700 base pairs. So it becomes too long to have all the combinations together and run it through a gel and then see the sequence. So it is not a very good idea to do longer sequences in Sanger sequencing. But this is actually uh, this is a drawback of other type of sequences as well. So for example, Illumina sequencing, the read length is around uh, including the insert size is around close to 700 base pair there also. So in Illumina sequencing, the most of the sequencing things that occur, most of the whole genome sequencing that occurs in today's world is done using Illumina sequencers. Those are the cheapest in terms of like sequencing the whole genome. Uh, but now we have technologies which can sequence like up to 10 kD, like 10,000 base pairs at once. But again, the issue with those are like those if your error rate increases with the increase in length of the sequence. So longer the sequence, more probable that you'll have error sequencing the one complete read together. So you- finding a balance between read length and quality of the bases that are getting sequenced is important. Right now, the technologies that are available, the short read ones include Illumina, and the long read are basically dominated by uh, Oxford Nanopore Technologies and PacBio. So they sequence up to 10 kb. Illumina sequences around 700, but uh, shorter read is easier and more kind of cost effective to sequence. Longer reads are much more expensive. Okay. Here also, but the thing is, uh, here also the issue is the limitation stems from the fact that read of length L of a sequence increases. Distinguishing the mass between L and L plus 1 becomes very difficult. So after you have done this half synthesis of these DNA strands, you need to run it through a gel. Now, as the length of the sequence increases, so it is easy to separate, is easier comparatively to separate a sequence of length 50 versus 55 probably, right? But it is much harder to distinguish between a sequence of length 1000 and 1005. So as your length increases, the difference in length, uh, identification of the difference in length and correctly identifying that makes it a much bigger issue. So yeah, so these are the limitations of Sanger sequencing. Next, we come something to called pyrosequencing. So this was early 2000s. This technology is in some ways deprecated, but yes, you'll find some papers use this till date. But yeah, mostly it is not used right now. So this was developed by a company called 454 Life Sciences, which was then acquired by Roche Diagnosis, and this is the pyrosequencing method using Roche 454 sequencing method. So the idea is that you have DNA inside water molecules in an oil solution, which will be an emulsion PCR. And inside each droplet, you'll have a single DNA template attached to a single primer coated bead, and which will form a clonal colony. And the sequencing machine contains this micro, the picoliter, not even micro, picoliter volume wells, like this one, what to say, uh, a plate, and inside the plate there will be like thousands of these micro wells in which the oil emulsification will be there, and uh, inside that the DNA strands. Right? Uh, and this pyrosequencing will use something called as an enzyme called luciferase, which will generate light for detection of the individual nucleotides that are added in the nascent DNA. Here also the idea is similar to sequencing by synthesis, that you're adding one base after the other to find what is the sequence of the whole DNA. So this uses some other technique to identify what bases are getting attached, and which is basically this enzyme, which is luciferase, which if you attach to the base, and when the base gets attached to the DNA, it'll give out some fluorescent light, and you can detect that using some machines, and you can know what base is getting added. Right. So yeah, this is the idea that you have this is your this is your original sequence that you want to sequence. Here is the DNA polymerase, here is the primer, and then you have DDTP. So basically, it's a base that is attached with some markers that when this base gets attached to this DNA, it'll release this ATP and sorry, AMP and PPI, and which can be used to convert back to ATP using ATP sulfurase. And this enzyme will be present in your system, and this will give out some light. So if you know and every base will have a distinct kind of signature for the light that it emits. So if you know what kind of signatures are corresponding to what kind of bases, you can detect this using some machine to detect the fluorescence, and you can get the whole sequence of the DNA. So this is also pretty pretty straightforward after Sanger sequencing. But now we come to Illumina. I'll not talk about in great detail today about the Illumina sequencing, but what I'll do is I'll give you a brief idea about how the sequencing occurs. I think I have a, just give me a moment. I think I have a good representation of that. Just a second. Yeah. So this is the link of the video uh, if you want to take a look about how Illumina sequencing works. I'll play it here as well and try to explain the things that are going on. Uh, s- yeah. So
The
the
Illumina sequence.
again, just start before starting, the idea is same as Sanger sequencing. The core idea is same, which is sequencing by synthesis. You have a single-stranded DNA. You want to add bases uh, on it to make it double-stranded, and you want to keep a track of what bases are getting added so that you can get the sequence, complementary sequence of the DNA. Right?
The Illumina sequencing workflow is composed of 4 basic steps: sample prep, cluster generation, sequencing, and data analysis. There are a number of different ways to prepare samples. All preparation methods add adapters to the ends of the DNA fragments. Through
So these are basically small stretches of the DNA to add to the end of the DNA so that you can have an unique identifier for the DNA, right? So these are acting as like identifiers, nothing
reduced
fancy.
cycle amplification, additional motifs are introduced, such as the sequencing binding site, indices, and regions complementary to the flow cell oligos. Clustering is a process where each fragment molecule is isothermally amplified. The flow cell is a glass slide with lanes. Each lane is a channel coated with alon, composed of 2 types of oligos. Hybridization is enabled by the first of the 2 types.
So these oligos that are present inside the wells, these are actually complementary to the adapters that are getting ligated to the sequence of your DNA, so that those can come and bind here. So that is the function of those
Types of
adapters.
oligos on the surface. This oligo is complementary to the adapter region on one of the fragment strands. A polymerase creates a complement of the hybridized fragment. The double-stranded molecule is denatured, and the original template is washed away. The strands are clonally amplified through bridge amplification.
So this is an interesting procedure that happens during Illumina sequencing, which is known as bridge amplification. Sorry. Yeah.
In this process, the strand folds over and the adapter region hybridizes to the second type of oligo on the flow cell. Polymerases generate the complementary strand, forming a double-stranded bridge. This bridge is denatured, resulting in 2 single-stranded copies of the molecule that are tethered to the flow cell.
This is the same copy.
The process is then repeated over and over and occurs simultaneously for millions of clusters, resulting in clonal amplification of all the fragments. After bridge amplification, the reverse strands are cleaved and washed off, leaving only the forward strands. The 3' ends are blocked to prevent unwanted priming.
Now, here is where the sequencing
Sequencing
occurs.
begins
So these procedures are basically just to amplify your DNA strands. Instead of using PCR and those kind of things, we use some more sophisticated techniques, clonal amplification, bridge amplification, these things. But the idea is just to amplify your DNA molecules. This is where the sequencing occurs.
with the extension of the first sequencing primer to produce the first read. With each cycle, fluorescently tagged nucleotides compete for addition to the growing chain. Only one is incorporated based on the sequence of the template. After the addition of each nucleotide, the clusters are excited by a light source, and a characteristic fluorescent signal is emitted. This proprietary process is called sequencing by synthesis.
See, the idea is same as Sanger sequencing. Instead, here you have fluorescent label tags with each of these molecules, and you can track in real time what molecules are getting attached. So, how if you see the output of a sequencing of a Illumina sequencer, it is basically kind of looks like a photo with different colors inside that. A black background, and on top of that there are different colors. So those different colors are actually the colors that are emitted while adding these pieces. So you need a software which is also called a base caller to identify these signals and convert them into bases. So the output of this signal of the sequencing is basically some signals, some light, fluorescent light signals that is getting emitted during the sequencing process.
The number of cycles determines the length of the read. The emission wavelength, along with the signal intensity, determines the base call. For a given cluster, all identical strands are read simultaneously. Hundreds of millions of clusters are sequenced in a massively parallel process.
Yes, that is the main. So this is how actually the output looks like. So you'll have a lots of these different colors dots, each color representing a particular nucleotide. Then you need a software to understand this whole image and the signal and convert it into different base calls.
This image represents a small fraction of the flow cell. After the completion of the first read, the read product is washed away. In this step, the Index 1 read primer is introduced and hybridized to the template. The read is generated similar to the first read. After completion of the index read, the read product is washed off and the 3' ends of the template are deprotected. The template now folds over and binds the second oligo on the flow cell. Index 2 is read in the same manner as Index 1.
So you see, you are reading the same read from one end to the other. So there is one read one, which is will be a 5' to 3' read, and there will be a read two, which will read the same exact sequence in an opposite manner, 3' to 5'. So that gives you a better confidence of how the sequence looks like in a particular region.
Polymerases extend the second flow cell oligo, forming a double-stranded bridge. This double-stranded DNA is then linearized and the 3' ends are blocked. The original forward strand is cleaved off and washed away, leaving only the reverse strand. Read 2 begins with the introduction of the Read 2 sequencing primer. As with Read 1, the sequencing steps are repeated until the desired read length is achieved. The Read 2 product is then washed away. This entire process generates millions of reads,
We'll not go into this. We'll look into the data analysis and our how we do it and those things. But yeah, this is the general idea about how the sequencing works. This is a modern technology. Illumina is used till date very widely, and it is one of the most cost-effective ways to sequence a genome, possibly human genomes. Now, the idea is that you have different cycles, and in each cycle you'll produce a combination of colors, and from that colors you can actually identify what read is getting, what actually the base at that position is, right? So just an overview of how it works again. So uh, the main characteristic of this type of sequencing is the massive parallelization that it can do. At one point of time, you can read so many sequences together. So that is the only that is what it has over Sanger sequencing. In Sanger sequencing, you can only read it at one at a time. You have to do it serially. But in Illumina, since you have such massive parallel lanes and so many so many wells inside those lanes that you can sequence a lot of reads together, right? So it's the idea is same that you have DDNTPs with fluorescent tags at this point of time. So you have fluorescent tags to identify what base is getting attached at real time, and you have a base caller that basically the pictures that get generated are analyzed by a base caller software to identify the basically sequence of the particular read. And for about for high-seq Illumina NovaSeq, the NovaSeq 6000 is basically the type of sequences that we use in the lab. But for high-seq, it takes about 6 minutes in a cycle. The NovaSeq 6000 is a little bit faster, a little bit more technologically advanced. That's all. Now, after you have done this whole synthesis using whole sequencing using Illumina, what you get is basically after the synthesis, you get that image kind of a file. Now, that image file will be passed through a base caller. The base caller will read those as particular bases in the DNA. So the output that you'll get is in the FASTQ format, which is the Illumina FASTQ format. I'll just quickly go over this. This is just technical stuff, like not very interesting, but a lot of information is stored here. So yeah, the FASTQ files will be .fq. I don't know if you have seen a FASTQ file or not, but it's readily available. If you want, you can just Google any FASTQ files. You'll get your hands on one. You download it and you can just take a look at how it looks basically. So for parent sequencing, it will produce two separate reads. As we saw, a single read is getting read twice. So you'll have uh, for one section of the DNA, you'll have two reads, right? From one side and the other, right? Not just like two reads supporting a particular portion of the DNA. It's like when that portion of the DNA is read, it is read from two ends. But you will have like 30, 40 such reads in one region of the DNA. So that is what is known as coverage. We'll come to that later. But yeah, just do not get confused with that. Yeah, so these two different reads will be usually stored in two different files, named in uh, one will be read1.fastq, the other will be read2.fastq, right? Uh, the fastq format is basically uh, four-line information, right? So you have whatever the number of reads are, your fastq file will have number of lines, which is a multiple of four, because each read information is captured in four lines, right? So the four lines, the first will be an identifier line, which will be a unique identifier for that particular base, for particular read. Then there will be the whole DNA sequence, ATGC, ATGC, the whole sequence. And after that, there will be a line that is almost always a simply a plus sign, but can be optionally followed by the repeat of the ID line. And after that, this is an important part. This is an ASCII coded Fed scale base quality score. So basically, it will give you for each base. So how have you gotten that base, right? First, you have done the Illumina sequencing, then you've gotten that image file, then you have passed it through the base caller. Now, the base caller, when it identifies a particular base from that image signal file, it it does so, but there is always a probability associated with it that it is doing it correctly or wrongly. So there is always a probability of getting that particular base wrong by the base caller itself. So what the base caller will do is provide a quality score of each of these reads, and that is displayed in your FASTQ file. So it's Fred scale, so it's basically log negative 10 log 10 to the power minus whatever. So if it is, say for example, your base caller does a mistake in identifying a base in uh, one every thousand bases, so it will be like negative 10 log of 10 to the power minus 3. So the Fred score will be 30 basically. So Fred score of 30 means the base caller does a wrong base calling one in every 1000 such bases. Now, if it is one in every 10,000, the Fred score will be 40. One in every 100,000, Fred score will be 50 and 60 and so on. So 99 is very high. So we generally do not have so high quality bases, but like 30 to 40 is the ideal range where you want your base quality as a minimum cutoff. Right. So this is a snapshot of how the Illumina file looks like. Second. Yeah. Sorry. So this is how the file looks like. So on the first line, these are your identifiers. So this is the identifier line. The second line is your set of bases for the read, whatever the sequence of the bases. Then it followed by your plus, and these are your ASCII coded quality values. So each of these elements, A or A for J or whatever, these are basically ASCII codes for different quality values. So you can convert them to quality values from ASCII back to your normal number system. So this is how the file will look like. So identifier, then the whole sequence of the read, and then the quality of the bases associated. So for T, the quality is A. For A, the quality is A again. So for example, here uh, for T, the quality is hyphen. So each of them actually correspond to a number in ASCII, which is telling you how confident the base caller is in actually identifying this particular base. So you need that uh, because if you do not have that, uh, you do not know what is the quality of the basis getting called. So you can do a, if you know the base quality, you can do a kind of a quality control. You can remove the bases that are falling behind below a certain threshold, and you can remove them. So that is important. So I'll just quickly go on the overview of the quality control of how we do this quality checks. Uh, I don't think all of this has been covered in your in the course itself. I just want to give you an idea about how in real life this data is generated and how we work with the data, right? So uh, once you have the FASTQ file, you need to do certain quality checks. So certain things. So for example, checking the quality of the base calls, as I was saying, to ensure there were no use issues during sequencing. Then you check the quality of the reads, quality matrix that adhere to our expectations. And then you also check for contamination. So this PCR duplicates that you're doing, this clonal amplification you're doing, the adapters that you're adding during Illumina synthesis, it can happen by mistake. The adapter also got sequenced, but that is not part of the data, right? So you need to identify these things and remove from your data set. So yeah, the QC goals include identifying sequencing problems, determining whether there is a need to contact the sequencing facility or how you have done the sequencing, identifying overrepresented contaminating sequences, gaining insight into library complexity if there is some RNA contamination or duplications of reads or whatever, and also every organism is represented by a certain GC content of the genome. So if you have like if you have a certain range of GC percentage of a particular genome, and if you are sequencing the same genome and you are finding a GC content very high or low, that means your sequencing quality is not very good. So probably you need to resequence. So that is also denoted by this QC part. So FastQC is a software that is used to do this. So I'll just quickly show you how it looks like. So this is the software. You can take a look. It's a very old software, but it is still used. It gives you a very good idea about how your data is actually. So it gives you all these plots, and yeah, it will give you what is the good data, what is the bad data, if you need to trim your adapter and those kind of things, right? So I'll give you an example of how it looks like. So It'll take FASTQ data, and it'll give you an HTML report of how the data is, what is the quality checks it has done, and what are the things that it has found. So yeah. So these are different types of things that it gives. So first it'll give a basic statistics of the sample. So generally, you have an idea about how the GC content should look like. So this is how it looks like. It'll give a table of basic statistics and total number of sequence, what is the median sequence length, what is the GC percentage. These are your basic statistics. Then you have per base sequence quality. So you have uh, maximum of these bases that are having low qualities, right? So this is not a good thing. So per base sequence quality is giving you the distribution of quality scores at each position in the read across all reads. So this plot can tell you whether there are problems occurring in the sequencing and where you are getting like more of these low quality basis sequence. So that's not a good sign. So you need to do probably your sequencing again, or you need to kind of kind of revisit the sequencing part. And yeah, uh, like that, it also gives per sequence quality scores as well. So that was per base quality score. This is per sequence quality score. This is how it actually looks like. So you have maximum of the bases on the distribution is skewed towards the right. So higher qualities, you have maximum of these bases, but you have also some low quality bases as well. But it generally should look like this, right? And this is the per sample per sequence GC content. It gives you a GC distribution of all sequences, but there is an expected GC percentage for the organism, which is given by this theoretical distribution. Sorry. Yeah, which is given by this theoretical distribution, and the red is basically your GC count per read. So it significantly deviates from your theoretical distribution, and that what it means is like there is some contamination or the sequencing has not occurred properly. So probably you need to revisit. And if there is some sort of overrepresentation, uh, with sharp peaks like this, which is indicative of some contamination or probably a very high expression of overexpressed gene. So, which is unlikely because that can only happen in probably disease cases or something out of the ordinary. But in a healthy situation, it should follow a theoretical distribution. Some deviation is okay, but this kind of deviation is not acceptable. Then you have these kind of information as well. So basically, sequence duplication levels it tells you, and what it gives you is an idea about the PCR amplification and is your reads contaminated with those PCR duplicates or not. So in RNA sequencing, we do not generally do anything for this analysis, but for the genome sequencing, for DNA sequencing, uh, if you have a very high number of these PCR cycles, this the contamination can occur like this. So these are your total sequences, the blue line, and these are your duplicated sequences, which is very high in number. So this is not ideal again, and it also gives you basically the overrepresented sequences. So you have, say for example, this particular read for 2,500 times, but it has no idea where the read is coming from. It is not actually in the DNA of the organism, but you have overrepresentation of that trait. It can occur that some genomes are very repetitive in nature, so you can have a read that is repeated multiple times. That is because of how the genome of the individual or the organism is. But in most cases, these overrepresented sequences are your adapter sequences or your PCR duplicates and those kind of things. So you can use BLAST to identify where the sequencing is coming from. You can do a sequence alignment, multiple sequence alignment, and you can try to see where is the origin of the sequence, right? So yeah, that is all I have in terms of discussion. Like I'll then now it's almost an hour. I'll take a couple of minutes. If you have any questions, or any doubts, I can answer that, and then we'll move on to some question solving. So this is what I how I like go about these classes are basically that I make PPTs on my own to kind of give you a little bit more on what the professor has already taught you in the course itself. So if you have any questions regarding that, also regarding the things that I just explained, you can ask me now, or else we'll take a couple of minutes break and then we'll move into solving the questions. The questions are not very difficult for today because this is the first class and like mostly it is theoretical questions, not much calculations involved. But yeah, if you want to ask anything, you can ask. Okay, if you do not have any questions, we can move on to the the questions. But before that, I would like to provide you with a couple of literature. Okay, there's a question. Sorry, I did not understand. Why do FASTQ files encode the base pairs as a whole ASCII character when it only requires 2 bits? Uh, so that is the whole point, right? Like if you do not want the whole values to be so those will take up much more greater spaces than the ASCII characters, right? So if you are encoding 40 or 50 or 60 with the ASCII character, the space required for the ASCII character will be lesser than encoding than using the whole number, right? I am not sure I get your question.
Sir, I mean for like the bases, they're like an ATGC, right?
Yeah.
So that each ATGC uses like I think 128 like 1 byte.
Mhm.
So instead of that, like I think they could have like allocated like 2 bits and then like use the rest of the bits to like encodes for like a partial Fred score or something like that.
Okay. So yeah, some so your logic is correct to a certain extent, but firstly, it can be done. It's not like you cannot do it. It's not that, but the focus of it is because since this was developed a little earlier, so the issue of human readability and simplicity is a point that you need to have certain some the value encoded in such a way that like even if you just look at the FASTQ file and you have a ASCII table, you can just easily interpret it, right? And it is also like kind of this binary format encoding would require more complex parsing mechanisms to for bioinformatics softwares to understand these FASTQ files. So it's more simpler to use ASCII coding, and it is more of a legacy thing than kind of like for our benefit that we are using, but it's more of a legacy thing that it has when it was started it went on like that. But the idea is that that you should be able to look at a FASTQ file and you could in a text editor and inspect it without any decoding. So for your cases, probably a little more decoding would be required, and it's not a very good idea to do that. But your point stands that yeah, it is true that we can use other type of encodings, and it is used in other type of files. So if you look at BAM files or CRAN files, which are basically this binary uh, uh, encoded format of these sequence alignment stuff. So those are those are uh, generally stored using these binary encodings, but you cannot, those are not human readable. But you cannot read the BAM file in a text editor or something like that. But a FASTQ file, it's more of a legacy thing. I hope I have answered your questions.
Yes, sir. Thank you.
Okay. Okay. So just let me share. Okay. So I think one thing that was discussed in the class was basically the T2T thing. Sorry. So as you know, the human genome was actually completed using this telomere to telomere consortium in 2022, 2023, where they sequenced the whole entire genome, which was previously not sequenced. There are several duplicated regions, repetitive regions, low complexity regions that were not being able to sequence earlier because it was very hard to identify where the reads are actually coming from. So in 2022, 2023, the T2T consortium actually sequenced the whole genome. So if you want to give it a read, this is one paper, and the other paper is, uh, if I'm not sure, T2T. So they did one for the X chromosome as well, one for the Y chromosome. This is more recent of 2023. So you can give it a read. These are very fascinating papers on how they use this technology. So for these, they did not use short reads. So they used PacBio Hi-Fi. So these are your ultra long reads, which are like around 100 kb. So that's a very long read that is getting sequenced together. Due to the advent of these technologies of this long read, we are being able to sequence the whole genome to understand the whole genome. So if you are interested in these kind of things, I'd suggest reading these couple of publications. I think you guys will like it. Okay. Uh, so then let's move on to the questions. These are simple questions. So I have not made up these questions. These are like older assignment questions. Some of them might be kind of overlapping with your current assignments. I do not have access to your current assignments. I only have access to the older assignments. So some of them might be overlapping with the current assignments. So I'm not sure about that. So if you have any issues with current assignment questions, I would I since I do not have access. So if you have any questions regarding that, you can send me to my email ID. Okay? So I'll take a look and let you know if I can help you with that. Okay, so yeah, so today's questions are not, I have not made it, but these are there in the assignment. So yeah, it's very kind of generic questions. You can put the answers in the chat box. Yeah, all of them. That's correct. Yeah, it's all of them. Phenomics is basically phenotypes. Omics is basically the overall term to describe everything but to a particular field. So for genomes, you have genomics. For RNA sequencing, so for understanding the gene expression, you have uh, you look at transcripts from the genes and you try to find which transcripts are getting overexpressed, underexpressed. So those are done using RNA sequencing and other microarray technologies. So that is transcriptomics. Epigenomics is basically finding the regions of the DNA where epigenetic modifications are occurring. So you have the DNA wrapped around histone noctamers. Now there will be certain places in that DNA where a particular methylation can occur or acetylation can occur. Depending on that, the histone can just kind of detach from that DNA at that position and kind of open up the DNA for replication, transcription, translation, whatever. So that is the base modifications are studied using epigenomics, and genomics is the study of all phenotypes. Yeah, that is true. It's the first generation. So Sanger sequencing is where it started. Then we have come today at second generation, which was Illumina sequencing. Third is basically the long read. Fourth is coming. I do not have very great idea about fourth generation anyway. Yeah, that is correct. The interesting thing about this is that this is the first protein to be sequenced, and it was sequenced is around like before 1960s by the same Frederick Sanger, and he won his first Nobel Prize due to this, the sequencing of bovine insulin, and later in 1970s he won the Nobel Prize for sequencing for the whole sequencing technology. But this was the first protein to be sequenced, and so uh, it was a huge deal back then. I'm not covered Edmund degradation, but I think Sir is covered. So yeah, So the answer is trifluoroacetic acid. There's another compound that is used for this amino terminal amino acid cleavage, which is known as PITC. You can search it. So I think phenyl isothiamate, if I'm not wrong. So there's another molecule. Uh, mm, yeah. So I think this is also used. PITC is also used for degradation. So if you want to know, that's extra information. Sorry. Just a second. Okay, couple of mixed answers. I'll wait a minute again. Okay. The answer is basically the first uh, option, which is the M2 protein. So this is a protein present on the coat of the bacteriophage. This was first sequenced around, I think, 1950s, if I'm not wrong. This is around like a 500 base pair long protein, not very huge, but decent sized protein. And this, sorry, decent sized gene. So yeah, this was the first. Okay, do we have any answers? B and D. Okay. Okay, so the answer is probably none of these. Yeah. So it is a simple unitary method. So you have 10 runs in a day, 105 reads in one run, and 1000 base pair per run. So total number of base pairs will be 10 times 105 times 1000, which is close to, I think, 1,050,000 a day. That is a throughput. Now, if you go from million to so million base pair to giga base pairs, so it will be close to, I think, yeah, 0.001 GBP per day. So none of this is correct. So it's nothing complicated. You just multiply all of them and you convert it to gigabases per day. That's all. So I think it's coming to 0.001 GB per day, GBP per day, which is none of these. Okay, moving on. Yeah, the answer is to 2023 because that is when this paper came out, the one that I showed you. So this this is 2023. This is around 2022. So around this time, actually, the whole sequence was determined. So there's 8% of the genome was remaining, which was sequenced during this time. So it's 2023. Okay. The answer is actually all of these. I'll quickly show you the ENCODE database. So this is actually a very well-maintained database. So you have a lot of these are all your projects that are going on that the data is for here. So if you go to the encyclopedia, so you have a lot of data here. So you have like these are all your epigenomic data, chromatin states, variant data, open chromatin and ChIP-seq data, RNA binding protein data. So there's a lot of data. It has gene data, it has protein data, it has I think others repeats, regulatory elements, everything. Mostly EQTLs is what we call. So this, if you see this page of ENCODE, this is actually what it has. So you have these fast queues, and from that you have aligned reads, and from that you can have so much information about DNA methylation, gene expression, ion binding proteins. Then you can have promoter information, enhancer information, uh, linked gene information, variant annotation information. So this is the whole basically encyclopedia for ENCODE. So if anybody is interested, please visit this page. And you'll get to know about ENCODE. This is actually one of the most used databases for genomic studies and mostly database work. So this is a very well maintained database as well. So if you are interested, please take a look. Okay. 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 Hmm, Mostly throughput and okay. The correct answer is throughput and cost effectiveness. So if you carefully think about it, the technology is not that massively different. It's the same sequencing by synthesis technology that was there in Sanger sequencing, that is there in Illumina sequencing. Illumina is the second generation sequencing, right? So uh, what has increased is, yeah, the massive parallelization that you can do using this kind of sequencing techniques. So the throughput increase is much high, and the cost effectiveness has gone down a lot since initial sequencing days because technologies advancements, uh, advances in chemistry, and so on. So the cost has gone down a lot, and it has gone down massively across, like, from the starting till today. It is we're looking at like 1/10 or 1/20th of the cost of sequencing a single genome compared to early 2000s, right? So less than 1/10, and 1/10 is a big estimate. It's probably 1/100th. So it has gone down massively, and throughput has increased again massively. So these are the two things that has happened. But the core technology is same between Sanger and second generation, which is sequencing by synthesis. Okay. Yeah, the answer is iron torrent sequencing and pyrosequencing. So, pyrosequencing, the Roche sequencing that I showed, that is one, and iron torrent sequencing is another, which is a very similar type of technology, and it comes under second generation. So, yeah. One thing I've not talked about, assembly and alignment. These two things I've not talked about today because I wanted to give a more general overview about sequencing and these technologies. I will talk about assembly approaches and alignment in the next classes. I'll go through the whole pipeline, the kind of pipeline we follow in lab to undergo, to kind of analyze genetic data in terms of sequencing data. So yeah, I'll go through the techniques of assembly and alignment in the future classes. But yeah. Okay, the answer is basically C. Someone answered it correctly, I saw. Optical map assisted genome assembly. But I'll not go into the details of this because I've not discussed genome assembly. But yeah, I'll discuss it in the next classes. But yeah, in the course lectures, I think Sir has discussed genome assembly to a certain extent. So yeah, I'll not comment much on this today. But yeah, this is the answer. Okay, so this is the final question. Just one second. This coverage matrix and all of these will come under alignment also. So I've not discussed that today as well. So discuss it in the future classes. See if you have any answers. Sorry, which problem is this? Arithmetic progression, geometric progression? Okay, can you please give me the question? I'm not sure which question you're talking about. Okay, for this case, the answer is B correctly. So the read number of reads is basically your coverage times your genome size divided by the read length. So your coverage is 49, your genome size is 3.2 into 10 to the power 9, and your read length is 200. So 49 times 3.2 into 10 to the power 9 divided by 200. So it comes around like I think 7.84 10 to the power 8. So a couple of people said D, which is correct. But yeah, can you please give me the question? I'm not sure which question you're talking about here.